Good evening, everyone, and a very warm welcome to the ninth monthly lecture series on scientists who lived so that humanity lives longer and healthier under the aegis of KK Biotech. And the scientist that we would be remembering today is Paul Ehrlich, who carried out outstanding work in hematology, immunology, and antimicrobial chemotherapy. So first, let me introduce our today's speaker, Dr. Abhay Patki, who is the Executive Director in Oncology Clinical Strategy and Development with Valo Health. Dr. Abhay did his BSc in microbiology, MSc in molecular genetics, and MPhil in molecular biology from University of Pune. He subsequently joined as a Commonwealth Scholar the University of Surrey for his doctoral research, where he studied the cloning and expression of mycobacterial genes for diagnosis and therapeutic purposes. He did a couple of years postdoc at Rush Medical Center, Chicago, before joining the Department of Medicine, Case Western Reserve University, Ohio, where he worked for close to 7 years. He is also an MBA from Case Western Reserve University, Cleveland. Since then, Dr. Abhay has taken leadership roles in various pharmaceutical giants that include GE Healthcare, Novartis Pharmaceuticals, Genmab, 10x Biopharma, Sanofi Pharmaceuticals, Blueprint Medicines, Radius Health, Surface Oncology, and Valo Health, where he is currently the executive director. Dr. Abhay is a highly experienced professional with extensive experience in global early and late stage clinical operations and drug development and due diligence activities. As a clinical scientist and an operations expert, he has led, directed, and managed complex trials to deliver successful results in immuno-oncology, hematology, oncology, neurology, and diagnostics using small molecules as well as biologics. He has been the clinical development team member of 6 drug approvals that include everolimus, daratumumab, ofatumumab, fedratinib, avapritinib, and ioflupin. He was a core clinical team member in 2 biotech companies that submitted 4 investigational new drugs, INDs, approved and successfully started first in human studies. He has got exceptional management, scientific, operational, problem solving, influencing, leadership, and teamwork skills. He has evaluated over 100 preclinical and clinical assets during due diligences, prepared presentations for go, no-go decisions, planning and preparation experience for INDs and NDAs filings. He has worked on joint protocol and research projects development and execution with partner companies, and he has produced over 25 scientific peer-reviewed publications and presentations. He is also currently a member of American Society of Clinical Oncology and American Society of Hematology. He has been awarded with several awards and honors. So it's a pleasure having Dr. Abhay with us and waiting for a very exciting lecture. But before we start that, I would like to hand over to Professor Kannan, the founder of KK Biotech, so that he can tell us something about KK Biotech, about this great initiative. Over to you, Professor Kannan.
Yeah. Thank you, Nathan.
And I'll take care of the slides.
Okay, thank you. Yeah. So it's a pleasure to have Abhay, who readily agreed to present today and in this exciting area. And I can see many of my friends, students, colleagues joining in. And especially I'm happy that Dr. Deepa Subramaniam at AstraZeneca has also joined in, she was one of the person who wrote to me, look, I want to attend this talk. I said, I'm very glad because she herself is deeply involved with this in AstraZeneca. So welcome Deepa and all my other friends up there. I can see my cousin Rajeshwari Iyer up there, who teaches in Canada in Toronto. So it's nice to have a mix of whole range of people, and I'm very happy that Abhay has agreed to join. The idea of starting, can you show the next slide?
Yes, please.
Yeah. So our aim was to bridge the gap between, in India, often what happens, there is a gap between the academic program, what is the real ground situation, and employability. And as you would have seen, the latest report from Government of India, uh, you will see that the university education is not providing employment. Number of unemployed are very high, and it's a small attempt by me to try to reduce the gap, at least in biotechnology. Okay, so that is what we are doing now, and we have been conducting a series of this series which we started about 9 months ago, and today Abe is speaking, and today's talk is dedicated to Paul Ehrlich, whom I have admired as one of the greatest minds of the century. Today's talk is, as I said, is dedicated to Paul Ehrlich. Next slide. Yeah. Oh. Okay. So we have also created a biotechnology education for students and teachers. The idea is that, this was a program which earlier we did it for biochemical education as part of the Society of Biological Chemists. Somehow it ran for about 9 years, then we couldn't continue it for various reasons. I thought that why not we restructure this so that students can know what is happening, because nowadays students are looking for employment or summer training. So I try to help them to find places where they can go and spend their time. Okay. We also, as I said, we provide online internship for students also, and also we encourage students, teachers, and postdocs to become member of our network so that they could interact with other people and have access to a larger this thing. Next slide. Yeah. So this is one of the programs which we did, which you can see in the website. So I won't like to spend too much time, but have a look at the website. You will come to know how the workshop was conducted. Now that again this COVID has started again, probably will restart this program, because in between students were too busy attending their classes in their respective universities. Next slide. Yeah. So, and as you know, many of them who conduct these workshops are officially paid, so that so it is so that there is students realize that the members who are presenting or the teachers who are teaching in the workshop are very committed, and they take a lot of their time out. And I must thank the first batch who conducted the workshop, and it was really a huge success. Thank you. Next one. Yeah. So today's talk, I just thought, I hand over to Abhay Patki to continue. Thank you, Nitin.
Thank you, sir. So I'll stop sharing the slides now so that Dr. Abhay can share, right?
Yeah.
Dr. Abhay, you may please start.
Okay. Can you see my slides now?
Yeah, we can see the slides.
Perfect.
Yeah,
Thank
please.
you. Yeah. So first of all, thanks a lot, Professor Kannan, for inviting me, and Nitin giving me a quiet introduction, nice introduction. So what I'm going to cover basically is the current status of novel checkpoint inhibitors that are approved, and a lot of them there are in the clinical trials. So I have divided talk based on this main theme. Obviously, it's a really, really large area, so it's not even possible to scratch the surface. But I have attempted to give overall 10,000 feet view so that we can at least get some understanding of checkpoint inhibitors, approved ones, and the ones that are in the clinical trials. So obviously, this talk is dedicated to Dr. Paul Ehrlich, and just to give a little bit of his achievements in the field of hematology, immunology, and chemotherapy. I have listed a few of his accomplishments here. The first one, I have highlighted mast cells in bold, because that is very dear to me, since I worked on one of the product for mast cells. In case of disease called systemic mastocytosis, there is increase of mast cells in the bone marrow, which are almost accounted for sometimes even 25%. And then there is a release of tryptase into the environment, which basically gives rise to disease called systemic mastocytosis. I have been fortunate enough to work on this disease at one of the company, and now the drug is approved for this particular disease, and patients are getting benefits. So that's why I have highlighted mast cell. Other his achievements, there is a long list in terms of hematology. When it comes to chemotherapy, actually, he was the first one to invent treatment for syphilis using various chemical compounds that he has produced. And one of the chemical series, the drug called Sarvesan was approved, and he was the founder of coining the term magic bullet. Last but not the least, very important for today's topic, is the concept of immune surveillance, was put forward by Paul Ehrlich almost 120, 130 years ago. So the field of immuno-oncology seems to be new, but really it's not new. A lot of work has been going on, but it has picked up a lot of space in last 20 to 30 years because of the various new discoveries. So definitely Paul Ehrlich played a really major role in immuno-oncology space right from the beginning. And he was awarded the Nobel Prize for immunology research in 1908. So coming back to today's topics and discussions, I have divided into 4 or 5 major topics. Very brief introduction of myself. I basically started my basic research graduation from Pune. Don't want to spend too much time. Nitin has already covered it. After bachelor's and master's, went to UK for PhD. Then came for US for first postdoc, second postdoc, and then since last 21 years, I'm in various biotechs and pharma companies, mainly based in New York, New Jersey, and Boston, Massachusetts area. I have extensive experience in the clinical trials. As you can see, I have done a lot of clinical research, clinical study concepts, protocol developments, and strategies, and really the execution of clinical trials and operations also in more than 20 trials and over 30 different countries all over the globe. And I've been really, really fortunate to work with really high-powered teams, which got approval in 7 different drugs. 7th one is about to come, but I have listed those here. Daratumumab, which is the multiple myeloma drug. I started the phase 1 study when I was in GenMab, and now it's over $4 billion drug. It's a first-line therapy for multiple myeloma, which is getting tremendous benefits for the patients who are living really number of years longer than what they used to live originally. The second contribution was for a drug called ofatumumab. This was also from GenMab. It was approved long time ago in CLL, and recently it got approved in multiple sclerosis. The third one was everolimus, also called Afinitor. It came from a company called Novartis. Everybody knows the company. I led the phase 3 trial for the approval of this drug in pancreatic neuroendocrine tumors, which is now yielding over $1 billion drug sales. And the few ones are like one in myelofibrosis. The last 2 that I mentioned are in GIST and systemic mastocytosis. These are highly specific precision medicines. So for example, this drug, avapritinib, is a kinase inhibitor. And in systemic mastocytosis, this one particular gene mutation from D8116 to valine is really the driver mutation for this disease. And the drug only acts on these particular mutants so that it shuts down this pathway and patients are getting benefit. And again, that was approved in last year in June. Hopefully, it will now be a worldwide product. Last but not the least, recent approval, the decision was made to submission for the drug called elacestrant. It's a SERD inhibitor. And when I was working at Radius Pharma, we started the first clinical trial. And the reason I'm showing is the one person or the person who are working in the clinical trials get immense satisfaction from the work that they are doing. And when the patients are first enrolled in the trial, and if they start to get benefit, you are the one in the clinical team are seeing the results for the first time. So here I have highlighted first 3 patients that were dosed using this drug, first in human studies, and all 3 patients got benefit even at the starting doses. So if you can see the picture here, the left panel basically shows the baseline of this patient when they were imaged using 18F PET, FESPET. And within 14 days of treatment, you can see complete or shutdown of the uptake of FESPET. That basically showing that the SERD is degrading all the receptors, and then later on these patients got benefit and their tumors reduced. So this is just the latest information that is coming out, and hopefully in 2022 this drug will be approved. Again, the reason I'm saying this because if there are students who are really willing to take the jump from academics to the biotechs and the clinical field, there are lots and lots of opportunities that one can make impact, and you can really do the basic research to the clinical, and the translation from bench to bedside is very, very satisfying. So a lot of opportunities outside academics are available for upcoming students who are finishing their bachelor's, master's, even PhDs, or even the postdoctoral fellowships. Now, coming back to immuno-oncology and today's discussion, here, very just brief overview that enormous R&D expenditure and research has been done. Lifespan of cancers patients have increased, but still it's a huge unmet medical need. Cancer is a really, really complex disease, multifactorial involvement of tissues, chemicals, compounds, pathways, etc. So, and its aggressiveness depends on the tissue type and stage of the disease. And still, the 4 major contributions who are accounting for majority of the deaths are prostate, lung, breast, and colon. There are a lot of other cancers, but these are the 4 ones that are major causes of deaths in the US. And I'm sure the picture is pretty much similar in Europe as well as rest of the world, including Asia. Now, initially, Hanahan in 2000 put together a concept that there are 6 hallmarks of cancer. Those are not highlighted here in the red lines. And later on, he upgraded that list from 6 to 8, then to 10. And recently, in January of this month, basically this year, he is now proposing there are 14 hallmarks of cancer. The reason I'm showing this slide, just to again emphasize that a lot of research has been going on, and there is a lot of new understanding that is coming out. So for example, now he is putting polymorphic microbiomes is one of the hallmark of cancer. Initially, nobody had even thought about microbes and microbiomes are playing any role in cancers. But now there is a lot of research that is going on, which is showing, and again, there are clinical stage companies who are producing drugs based on the microbiomes. Another example is understanding the plasticity or senescent cells, just to show that the number of research experience and expertise that are ongoing is really, really huge. And obviously, immuno-oncology is one of the major driver and sucking up a lot of resources, rightfully so, because I'm working on it. So that's what could be my bias. But again, just to show that it's not the only important factor, there are a lot of other factors also that basically causes the cancer. Coming back to really basics of immunity and cancer, there are two basic mechanisms, defense systems, innate and adaptive. Adaptive is again divided into humoral and cellular, antibodies versus T and B cells. And then really the interplay of immune system and cancer is based on 3 E's. Either cancer cells are getting eliminated, or they are in equilibrium. That means the disease is stable. Elimination is basically cancer is gone. And if the cancer cells escape, then the disease progression happens. So these 3 E's are really important basis of immune surveillance and immune mechanisms. So to understand the immuno-oncology, one has to really understand the hematology first. And that's the reason why I put this slide. There are obviously bone marrow and progenitor cells. And the cells that are involved in innate immunity are marked by green. For example, neutrophils. And cells that are actively involved in adaptive immunity, mainly T and B cells and NKs. And there are, of course, other cells which also play, like macrophages, dendritic cells, mast cells, etc. So these are the basic cells that are really involved in immuno-oncology and are responsible for either elimination of cancer or cancer escapes because of the various mechanisms that are coming out. Coming back to common types of cancer immunotherapies, the first and foremost immunotherapy that is approved for IO field, I would say, is IL-2. And IL-2 was approved in late 90s in RCC and melanoma. And the response rates were in the range of 18 to 20%. Still, 80% patient did not respond. But even who responded, 18 to 20%, those patients had really nasty side effects. For example, cytokine storm was pretty common. Endothelial leakage was the major reason why it was not readily used. And the reason for those side effects are IL-2 basically activates all T cells, including Tregs. And Tregs are really important because those cells are, in case of IL-2, were causing a lot of side effects. And the reason I'm highlighting IL-2 again is currently I'm working on the modified IL-2, which basically will not bind to Tregs, and it will selectively expand only CD8s, CTLs, NKs, and gamma delta cells. So basically, it's improved IL-2 using the really cool technology, which we will not go into it. But that's just one example that a lot of research is ongoing. The other types of, obviously, everybody knows about CAR T cells, which are again changing the field. Today, we will mainly focus on CTLA, PD-1, PD-L1, and some of the other checkpoint inhibitors like TIGIT, LAG3, OX40. The list is growing tremendously day by day. I will also focus a little bit on adenosine pathways, where the small molecules are involved for inhibiting A2AR and A2BR receptors. And then there are other types of immunotherapies like vaccines, which are also in the clinical trials with some and limited success. And obviously, there are a lot of cases where treatment combinations are being used to treat cancers. The concept of hot and cold tumors, I will introduce slowly in the next slide or so. But here, the purpose of this slide is to show that there are different drugs and combinations that are ongoing. And you can see the type of therapies and approaches that various companies are taking in terms of combinations. So T-cell targeted immunomodulators, for example, IL-2 and a lot of others are being looked into either hot tumors or cold tumors or a lot of altered tumors also. So on and so forth. We don't have to go through this list one by one, but suffice to say that a lot of activities are ongoing. Now coming back to concept of tumor microenvironment, TME, and hot and cold tumors. So basically, based on the invasion of tumor cells, based on the invasion of T cells, NK cells, or the immune cells, tumors are divided into 4 major types. The first one is hot tumors, where there is a high degree of T cell and CTL infiltration. It's also called immunoscore. So immunoscore for these hot tumors is really high. And the checkpoint activation, a lot of work is ongoing for PD-1, PD-L1, CTLA-4. And the first approvals that came from checkpoint inhibitors are coming from tumors which are hot, i.e. melanoma, non-small cell lung cancer. The second immunoscore type is altered immunosuppressed immune tumors, where there is a poor or less T cell infiltration, where soluble inhibitory mediators are present. And there are some cells which are responsible for immune suppression, like MDSCs and Tregs are also seen in these type of intermediate immune tumors. Altered immune excluded tumors, basically there is no T cell infiltration. So in these patients, if you give PD-1, PD-L1, there is very less chance that those drugs will act in these tumors. Basically, the tumors are cold, so there is no infiltration of cells. So PD-1, PD-L1 may not act. And there is a lot of research ongoing basically now how we can convert these cold tumors into hot. And it is possible to convert these cold tumors into hot by various mechanisms. And once these tumors are converted into hot tumors, then PD-1, PD-L1, or CTLA-4 will be acting. And I will show in some of the data from this concept in one of the future slides. And the last one is obviously cold tumors, where there is basically zero infiltration of T cells, NK cells, CD8s, CTLs. And this is shown very nicely in this particular pictorial slide. Here on the top left, you can see a hot tumor, which is completely filled with CTLs, NKs, CTLs, gamma deltas. On the other hand, if you look at the top right corner, it's a cold tumor. By immunophenotyping, you cannot detect any T cells whatsoever. Here, altered tumors. So here you can see at the edge of the tumor, all the T cells, NKs are concentrated. And here, altered intermediate immunoscore, where there is some infiltration of T and NKs into the tumor. So the purpose of research is how we can convert these 3 tumor types or tumor microenvironments into the hot tumor type here. And here again, I have basically said that if the tumor is hot, then the checkpoint inhibitors work really well. As the tumor gets colder and colder, there is no activation of CTLAs and NKs. And again, the use of combination, now it is possible, but still a lot of research has to be done. But there are some success stories where scientists have converted cold tumors into the hot tumors. This is again a really nice Nature review that I pulled together slide from 2019. So basically, this is the tumor divided into cold, hot, excluded, and immunosuppressed by the second concentric circle. Then the circle in green basically tells a little bit about which are the different pathways that are involved in oncogenesis of these tumors. The next concentric circle, blue, is giving information about various pathways. And then the last outermost circle gives the actual targets that are being looked into these various cancers and types. And these are the ones that are currently approved drugs in various cancer types or various pathways. So anti-PD-1, PD-L1, CTLA, you can see are mainly focused on hot tumors. Then the IL-2, interferon alpha, some of the kinase inhibitors like mTOR, various TKIs for various EGFR or specific tumor mutations for non-small cell lung cancers, cartel therapy, anti-CTLA combinations, oncolytic viruses, TKIs, and radiotherapy. So the purpose of this is to show that there are a lot of targets that are actively being looked at by various companies, various clinical stage research organizations, also academics, obviously. And academics are producing, again, a lot of data to push out new targets, which will be seen by chemical entities. And then the pharma biotech can pick up those targets and develop drugs for those. The main basic biological rationale of immune checkpoint is like this. Analogy you can give as car driving. So to start the car driving, you have to release the brakes. But just by releasing brakes, car will not move. You have to give the accelerator also. So you have to give the gas. So the way that a lot of people are describing immune checkpoint inhibitors is you are releasing the brake, but also at the same time starting to giving a lot of gas so that car can move and immune cells can get activated and basically will start driving the immune system and elimination of cancer. So here on the left-hand panel, you can see the blockage of CTLA-4 by anti-CTLA antibodies. That will basically activate the system. And then if you add PD-1, PD-L1 inhibitors in combination with CTLA-4, that will basically block the MHC interaction or the PD-L1 PD-L1 interaction. The TCR action interaction with MHC will still go on, and that will release into activation of T cells. Activated T cells will start their job of releasing other cytokines, chemokines, engaging other active cells into the TME, and which will basically kill the tumor. So this is the basic concept of immune checkpoint inhibitors by releasing the gas and giving the gas and releasing the brake. The really important paper came in 2013 about the somatic mutations. So this study was done using over 7,000 patients from various cancer types, almost 20 cancer patient types. And it was a large collaboration where over 5 million mutations were identified. And basically to show that the mutation rates in each cancer type differs highly. And that is the reason why you are seeing melanoma lung cancer in the bottom on the right-hand corner here, to show that the high prevalence of somatic mutations, that means there is a high tumor burden, high mutations rate. That's why the PD-1, PD-L1 will work, because of the engagement of MHC complex is really high in those high tumor mutation burdens and high somatic mutations. This was the important paper which led to a lot of explosion in R&D for immuno-oncology field. So in this part of first summary, I can say that I have convinced you that cancer is still a high unmet medical need. Understanding of immuno-oncology is really depending on understanding of the immune system, mechanisms, and immunity. Cancers are heterogeneous, as we just saw from the previous slide, and understanding tumor microenvironment is really, really important for understanding immuno-oncology checkpoints. In the next topic, I'll briefly discuss about the status of checkpoint inhibitors. Just a milestone of immuno-oncology, I just mentioned that IO is not new. It's more than 130 years old. A lot of progress have been made in last 30 years, mainly because of the development of identification of CTLA-4 and PD-1. But the real work, I can say, that started in inflammation and cancer by Rudolf Virchow in 1863, where he identified leukocytes as one of the major concern of inflammation and connection between inflammation and cancer, followed by Paul Ehrlich. We just briefly touched upon his accomplishments. The major breakthrough came in identification of monoclonal antibodies, in 1975. Again, these three got the Nobel Prize for that. And the first monoclonal antibody was approved for NHL in 1997. That was rituximab. And that basically started the whole explosion of monoclonal antibodies research and approval after 1997. The major breakthrough again came in 90 and 92 by identification of CTLA4 by James Allison in Texas and Honjo in Japan. And that led to the first checkpoint inhibitors approval for ipilimumab, Opdivo, and pembrolizumab, which is also called Keytruda for melanoma in 2014. And EP was first approved in melanoma in 2011. So this is just a small attempt to show the progression of immuno-oncology. And now, if we turn to 2022 or 2021, there are 100 monoclonal antibodies that are approved in various fields. Out of those 100 monoclonal antibodies, 7 are classified into PD-1, PD-L1. 4 are approved for PD-1. The 100th one was the PD-1 approved by GSK recently, last year. And there are 3 anti-PD-L1 that are approved. As you can see, the first approvals in 2014 and 2011 came in the patients without any biomarkers. So there was no bone marrow biopsy needed. There was no companion diagnostics. So that's why the response rates were really, really low. And as the science progressed, patient selections became really important. Patient biomarkers are becoming really important. And you can see now lately, there are a lot of approvals that are coming specifically based on the selection of patients, which are based on the biomarkers, or there is a companion diagnostics, which is a part of the approval for selecting the patients based on that companion diagnostics. Really important slide here to show that how immuno-oncology and some of the drugs, especially in melanoma, have changed the lifespan and impact on patients. Here, the bottom left or the left slide basically shows the survival rate before 2011. 50% of the overall survival is less than 6 months in these patients for stage 4 melanoma patients. And this is data collected from 42 various phase 2 trials. Again, the stage of the disease is stage 4. But when Pembro was introduced, along with IPI, the rates started to go up dramatically. And this data is still maturing at that time when the slide was put together. As you can see, 2 years, the death rate, overall survival, is really, really high. It's almost like 85 to even 90%. And fast forward in 2021, now we can see the same trials where they continue to monitor patients. One can see the overall survival rate for 50% is still over 6 years. So these are 72 months, 75 months data. If the patients are getting a combination of Nivo and IPI, 50% patients are still alive at even 6.5 years. So this shows the power of immuno-oncology. Again, these are limited number of patients. Again, I'd say that only 30 to 40% patients respond, but patients who respond live longer and longer. So still a lot of need to improve because 60 to 70% patients do not respond. And researchers are trying to find out why these 60 to 70 patients are not responding and how to increase that response rate from 30% to 50, 60, or even 80%. And eventually patients who progress, how to stop that progressive disease. Even if you try to maintain the disease as a stable disease, still that's a win for the patient because patients will live longer. Again, one more slide to show that this data is coming from patients who have metastatic non-small cell lung cancer, as compared to chemotherapy, where overall response rate, overall survival was 13.4 months median, versus patients who are on pembro, we have doubled the overall survival rate from 13 months to 26 months. Really, one thing to note for clinicians is the concept of pseudoprogression. So what happens with the introduction of immunocheckpoint inhibitors is once patients start to get the drug, cold tumors become infiltrated with the T cells and NK cells. So if you just do the imaging of these tumors, on the tumor imaging studies, the tumors show that the tumors are growing. So initially what clinicians thought that the disease is progressing, and they take out those patients from the trial immediately just based on the progression of the disease based on the tumor. But later on, as the research went on, they realized that it was a pseudoprogression, and the term pseudoprogression was coined. And roughly in even between 9 to 20% patients, this pseudoprogression happens. And the important lesson that clinicians learn is they should not take patients off the drug just because the tumors are progressing based on the imaging. And that's how the concept of, there is a concept called RESIST, which is a common terminology to measure the tumor in clinical trials. That is now modified for iRESIST. That means if there is an immuno-oncology trial, then the iRESIST criteria should be used, where if the tumor progress based on one time point, and if at the second time point, which is 4 to 8 weeks, if the tumor is still progressing, then the patients are considered as, okay, they are really progressing. And if the second time point is seen that tumor is shrinking, then it's a pseudoprogression, and the patients should remain on the study, and they will get benefit later on. Coming back to a little bit of non-small lung cancer now. Initially, just based on the disease criteria, based on the histology, it was just considered as one type of disease. Then later on, it was divided into squamous and adenotype. And now it's really a growing field. And based on the mutation of the disease, it's highly divided into specific mutations of the disease for non-small cell lung cancer: EGFR, MET, HER2, ROS, BRAF, RAT, NTRK, PI3K, MEK. The reason I'm showing this because for all these specific mutations, now there are companies and drugs that are being developed, some are approved, which are specific for these particular kinase inhibitors. And for these patients, PD-1, PD-L1 or checkpoints are not given as a first-line therapy. They will receive those specific drugs as a first-line therapy. For example, here, if the person is having ALK mutation, they will get ALK-specific kinase drug, crizotinib, alectinib, ceritinib, etc. If the patient is having EGFR mutations, they will get specific drugs that are approved for EGFR. And that's why the characterization of cancer type is really, really important. So when the patient is diagnosed as non-small cell lung cancer, immediately patient should get further characterization of the tumor itself. So for example, now Foundation Medicine has a cancer panel of 5,000 genes. So the tumor biopsy is done, and those tumor biopsies are sent to Foundation Medicine panel. Specific mutations are identified, and if the specific mutations are driving that driving that cancer, then those drugs are given. So for example, RAS or ROS, recently drug is approved, or NTRK fusion, the recent 2 drugs that are approved. Again, these patient numbers are really, really small, but if they are not given proper treatment, if they just get general chemo or PD-1, PD-L1, they will not be benefiting from those drugs. they should be given a specific targeted drug for targeted mutation. But now, when those patients fail, eventually patients ultimately fail even though they get targeted drugs. And now the research is going on. If they failed those targeted mutants, can they come back to PD-1, PD-L1, or CTLA, and will they get benefit? And yes, there are now data that is coming on, and those patients are now put on PD-1, PD-L1 therapies, and they do get benefit. But again, there are some nuances where they're looking for tumor mutation burden and whether there is a presence of PD-L1 on the tumor. So here one can see if the tumor mutation burden is high and if the PD-L1 expression is also high, then there is a pretty good chance that they will get benefit of PD-1, PD-L1. And as the tumor mutation burden goes low and the PD-L1 is less, then usually they will not respond to the drug. Again, shows the benefit of biomarkers, companion diagnostics, and understanding the tumor type at the molecular level, not just based on the gross biopsy or just based on the imaging. I briefly mentioned that the tumor mutation burden and the seminal paper that came from Nature in 2013, which shows the somatic mutations play a really important role in understanding. Here it shows the relationship between the objective response rate and the tumor mutation burden. So if the tumor is highly mutated, as you can see from the top three here, is cutaneous squamous cell or colorectal cancer or melanoma, the high mutation burden, the objective response rates are high for checkpoint inhibitors. And as the mutation burden goes down, for example, pancreatic or juvenile carcinoma, checkpoint inhibitors may not respond because the tumor mutation burden is really, really low. So one of the clever things that companies are doing is selecting the patients based on the specific biomarker. So for example, what Merck did for Keytruda is they did a combined study of patients who had, in the first case, tumor mutation burden was high. So it was an agnostic trial where they pooled data from multiple trials, analyzed tumor mutation burden, and looked where the response rates are high or not. And they found out that yes, if the tumor mutation burden is greater than 10 mutations per megabyte, then the response rates are really high, 29%. And based on this particular analysis from very small study, only 102 patients, single arm, phase 2 study, they got the approval. The second approval also came from same company for Keytruda for MSI high patients, where they basically looked at data from 5 different uncontrolled trials, only 150 patients, and again single arm trials, but they show that the response rates were really, really high if the patients are having MSI high or DAMMR as a type of tumor. Just to prove that initially the clinical drug development was really complex. People were doing traditional phase 1 study, phase 2 study, large phase 3, double-blinded, 2 controlled clinical trials, which range in the range of 300 to 600 or even 1,000 patients. But now the clinical trials is really, really different. Sometimes, based on the phase 1 study itself, regulatory agencies are willing to look at the data and give approvals. Another example, which is not coming from the IO field, for KIT inhibitor that I talked about briefly for Blueprint Medicines, the drug approval came based on only 42 patients in systemic mastocytosis. Or in GIST, it was roughly 50 to 60 patient study, because that patient population is so small that FDA has realized that if you try to enroll 300 patients, it will take 10, 15 years. And by that time, patients are not going to get benefit of this potentially good drug. And they're willing to understand and listen to the new data and give approvals based on limited number of patients. Now, there are a lot of reasons why patients are becoming negative, or even they're failing. Some of the reasons are if the biopsy is not right, if it is too small. The biopsy location is really important. The tumor is heterogeneous. So which type of tumor location that you are using to take the biopsy, and unfortunately from that particular location, if you get the biopsy where T cells are not present or the PDL1 is not there, then it will give the wrong information. And again, the timing related to therapy is also important, because some of these markers are dynamic. Or if the target is internalized, or if not present on the cell surface, and at that time, if you get the sampling, you will get the wrong results, and you may not get the right therapy for that patient. And again, there is a lot of mutational burden diversity and new antigen diversity also. Now, in terms of the side effects, obviously, no drug is safe. Even the water is not safe. There are side effects of each and every drug. And the side effects of checkpoint inhibitors are slightly different than the previously experienced side effects from other drugs. Here, one of the slides that I'm showing is attempting to basically give a little bit of information about what type of side effects are being seen. Some of these are grade 1 and 2. So these grades are defined based on criteria that are defined by CTCAE criteria. And patients, if they are experiencing grade 1 and 2 side effects, these are considered as minor, and patient can tolerate because the benefit is really, really high. So there has to be a balanced ratio of safety to benefit. But if the grades are increasing between 3 to 4, then obviously there has to be intervention. Grade 5 is obviously not good because patient is dead. That's what defined as grade 5 AE. But obviously there are a lot of grade 3 or 4 events, but those has to be managed. And those are different than the traditional chemo side effects or traditional other drug side effects. So the physicians who are treating these patients with IOs have to understand and be really tuned in to understand and development of new side effects that are coming on based on the data that are coming out of these patients. So just to summarize this part of the talk, IO field is exploding. Definitely progression-free survival and overall survival rates have increased. Still, a lot of patients do not respond. 70% patients still are not responding. Now we have to figure out how and why these patients are not responding and how to increase that response rate. Patients who respond eventually fail. Again, a lot of research has to be done why they are failing. And a lot of understanding is needed. In the last part, I will just try to focus some of the future directions for new checkpoint inhibitors. As I briefly said, tumor microenvironment is really important to understand. What are different new targets, interactions that are happening between the tumor cells and the CTLs, the dendritic cells, the macrophage population is also really important. There is a lot of conversion of M1 to M2 macrophages. Fibroblasts, which are present in the TME, are really important in terms of understanding the T cells. And the TH1-TH ratio, Tregs, again, the presence or absence of Tregs is playing some role because they are dampening the immune response if the high Tregs are present. how we can get rid of Tregs from the TME or lower their response rate is also important. And based on these, there are a lot of targets that are being studied. Some of these targets are cell surface targets, and a lot of other targets are internal targets. Obviously, monoclonal antibodies will not work on internal targets where small molecules will come into play. And that's why the combination of small molecules and MABs is, again, important to basically block the pathways at two different levels, one at the cell surface level and one at the internal cell levels. Some of the top monoclonal antibody targets I have shown here, and a number of investigational studies that are being done. Obviously, the winner is PD-1, PD-L1. And then there are a lot of other targets that are mentioned here. Some of these, again, studied as a monotherapy. Some are studied as a combination therapy. I also worked at my previous company on CD47, which is a really hot target nowadays. It's called a do not eat me signal, or eat me signal, the way that you look at it. It's highly promising target based on the data that is coming out of Gilead. It's now initially studied in hematology indications, but now the work is expanding in solid tumors also, in combination with various other agents. This again gives some of the targets based on the T cells. Obviously, the first T cell signal is MHC TCR interactions, and the top signals are T cell activation signals, and the bottom signals shown in red are signals that are inhibiting T cells. And obviously, the combination of cytokines is also important to manage the interactions of T cells, tumor cells, and other interaction T cells, macrophages, Tregs, etc. To give a little bit more information on the markers on the cell surface versus intrinsic pathways is depicted here. And then these are the two targets where drugs are approved, and these are the cell surface targets where a lot of ongoing clinical trials are getting some promising data sets. So for example, LAG3 and TIGIT are initially really hot target. Now BCA1H3 and TIM3 is becoming hot. So depending on the data that is coming out of phase 1 and 2, the field is changing. The combination studies are coming. The data is still coming. It's still in infancy, but a lot of promising data is coming out. And in the last few slides, I'll focus a little bit more on the adenosine pathway or adenosine axis. And the reason I selected this pathway is just to show the importance of interactions that are happening between the tumor cells and other cells, which are like NKs and CTLs. So what happens in the tumor based on the release of ATP, that ATP gets released by tumor cells and it gets converted into AMP by CD39, and then AMP gets converted into free adenosine by CD73. So based on this pathway, initially people thought that, okay, in that case, if you can just block by a monoclonal antibody against CD39 or monoclonal antibody against CD73, you can reduce the adenosine level in the TME. And if the adenosine level is high, what happens? The adenosine is taken up by T cells by using A2AR and A2BR receptors, which are present on T cells and NK cells. And basically, if these receptors are active, if they take up adenosine, NK cell cytotoxicity is decreased. And also for T cells, T effector function and cytotoxicity is decreased. And in terms of myeloid cells also, the T cell activation is decreased by uptake of adenosine. So here one can think of three different but distinct, but at the same time having similar impact drugs. So one can look for antibodies for CD39 and CD73. And yes, a lot of companies are working on these antibodies. Phase 1 studies are ongoing. Or one can look at the small molecules inhibitors, which will basically stop this receptor, and then free adenosine will not be taken up by NKs, CTLs, and myeloid cells. That way, you will see still increased activity of T cells and NK cells. And in subsequent slides, I'll show you some of the data for these particular three inhibitors. The company called iTeos is working on a small molecule which will block this uptake of adenosine by blocking this A2AR receptor. They have developed this highly efficacious molecule called enopidant. So if you look at the adenosine concentration in a normal cell environment, it's less than 1 micromolar. And in the tumor cell, the adenosine concentration goes up really, really high. It's sometimes even 10 to 20 micromolar concentration of adenosine is seen in the tumor microenvironment, which is really, really super high adenosine concentration. So obviously the drugs, there are like 4 or 5 companies which are working on these small molecules which will block this A2AR. And those are shown here. One is from AB920. AstraZeneca is working on a molecule called AZD4635. There is a molecule from Novartis, 178. And there's a molecule from Corvus called CPI444. And then the top one here is from iTeos. And the reason I'm showing this slide is when you look at the potency of iTeos molecule, whether in the tumor microenvironment is really, really low at IC50 of even less than 1 to 2 nanomolar, this drug is highly active in the TME, whereas other molecules are active, but one has to look at the IC50, it's in the range of micromolar. So based on this, one can see that enopidant has really, really strong potential and may be the winner in this particular race of small molecules from these 5 different drugs. So this study came irrespective of the drugs, but what they did is looked at the adenosine high versus adenosine low signature on the tumors. And this is all tumor study, not specific to any particular indication. But one can see that if the adenosine levels are high, the overall survival rate is substantially low. If the adenosine levels are low, the overall survival rate is a little bit better. And the same thing holds true when you look at the right-hand panel, which shows the progression-free survival. Again, if the adenosine levels are high, patients progress relatively faster, and this is a significant difference. If the adenosine levels are low, the patients progress slower, as seen in gray. Okay. So based on this, now what companies are doing is they're trying to do the patient selection based on these biomarkers. So for example, this slide I borrowed from a company called Arcus Biosciences. They have done a lot of studies in various tumor types. The first study is here listed in non-small cell lung cancer. And again, they are trying the combination approach of their molecule in combination with pembro and chemotherapy. And there they have shown the response rate, overall survival rate is 31%. The stable disease is 46%. Again, this is a lot of improvement as compared to the patients who have basically relapse refractory and they do not have any other options left, where they progress really fast. Here, if you look at from same ARCUS, look at metastatic cancer for colorectal in combination with FOLFOX, the response rate in the first-line therapy is 53%, and 43% patients are showing a stable disease. Again, the numbers are really small, but yes, there is a lot of promise. And even in the third-line patients, where they have failed first 2 lines, nothing is available, they are relapse refractory, overall survival and PFS is also promising here. We are doubling the survival rate in these patients. And again, the same company, when they did the second-line patients in prostate cancer in combination setting, the response rates are remarkable. Not very high, but definitely moving the needle in the right direction. Another company called Corpus, they tried to do gene signature. And what they did is, again, looking at, this is data from baseline to after treatment, whether the patients are IO naive or prior therapy. Not that great, but again, if you look at the combination, then you can see monotherapy versus combination, the drugs are showing better survival probability and better progression-free survival. Again, the power of combination is pretty high here. And here the example where they have shown, again, a proof of concept study that the cold tumors can be converted into hot tumors. This is the example where they have shown by doing the biopsy pre and post treatment. Here the pre-treatment, you do not see, or you see very little infiltration of CTLs. And after the post-treatment, you can see a really good infiltration of CTLs or CD8s. That means you have converted your cold tumor or intermediate immunoscore tumor into hot tumor. And once you convert that cold tumor or intermediate tumor into hot tumor, then you can come with a combination therapy approach. And when you give atezolizumab here, now patients will start responding. The concept is really cool here. And this is, I think, one of the first data where they have clearly shown the conversion of cold tumors into hot. And again, the same company is looking at the signature series from biomarker sets. So basically, this is a retrospective study. What they did is, after taking pre and post biopsies and looking at the overall signature, and then corresponding that signature to the response versus non-response rates. One, you can see that based on the signature, now they looked at the response rates. And in the panel B, you can see if the adenosine signature is low, patients did not respond. They either have a stable disease or progressive disease, whether they've given monotherapy or combination therapy. But if you look at the signature where the adenosine signature is high, now you can clearly see the response rate is increasing. And again, even in monotherapy setting, the patients were responding, but obviously when the combination was given, the response rates were really high. Again, to show the power of research, the biomarker strategy, and developing a specific signature pattern for patients before you start the treatment. And again, here they have seen the survival rate with a really high p-value for adenosine signature positive patients. The survival rate is better as compared to adenosine signature negative patients. So I think in summary, you can say that there are a lot more targets that are being still studied other than PD-1, PD-L1. Obviously, a lot of research is going on in combination studies. Understanding of TME for patient selection is really important, and also for the drug selection it's really important. Combination therapy will be going forward major approach. Monotherapy is not going to win, and personalized medicine approach is really promising, and a lot of research is going on towards personalized medicine based on the biomarkers and signatures. And I think that's my last slide over there. Thank you.
Thank you, Dr. Abhay. Thank you for the highly interesting and informative lecture on immuno-oncology. And it seems the biologics are the way forward, even though there are research activities going on with the small molecules. But to me, it appeared as if the biologics has an upper hand, the monoclonal antibodies. And it must have been very satisfying to be part of the development of drugs that could ultimately reach the patients and save the precious human life. I would now like to open the session for discussion. And I think there are some questions in the chat box itself.
Yeah, why don't you read it out, Nitin?
Yeah, yes, yeah, sure.
Most of mine are mostly comments made real time as Dr. Abhay was talking, just to add to the topic, not really questions.
Yes, yeah.
I think just read it out for the information of others, because everybody may not have seen the comment. Why did you quickly go through the comments of Dr.
Sure. The comments are these, yeah. The response rate is variable between different.
Nitin, I'm not sure you need to read through every comment. I think it was really made in real time as Dr. Abhay was talking. I think the concepts are essentially, I think he touched upon the immune cold and the immune hot tumors. So a large amount of the effort right now is to try to convert immune cold tumors into immune hot. So he was talking about the adenosine pathway. It's a pathway I've also worked on. As Kannan mentioned, I am working at AstraZeneca. I'm a clinician, clinical oncologist, who's been in the clinic for about 17 years at Georgetown before I moved to the industry just a little over 2 years ago. But the first drug I worked on was actually a CD73 monoclonal antibody that just entered a phase 3 trial. And right now I work on immuno-oncology. I work on more classical checkpoints right now. Some of I can't really talk about it because that data is yet to be published. It should be coming at AACR, our first publication with the molecule I'm working on. I'll talk to you about it after that. So I think I just mentioned some of the other tumor types. I'm not sure of the nature of the audience here. If most of you are biotechnology background, I thought it would be helpful for you to understand what are the tumors where we are desperately trying to make progress. I think colorectal cancer, or more commonly called colon cancer, is a tumor where we've really been struggling. If you take colon cancer, you can divide it into two main subsets, MSI high, and which is known as microsatellite instability high. And then the other group, which is actually the more common garden variety colon cancer, has what is known as microsatellite stability, or MSS colorectal cancer. MSS colorectal cancer is the most common kind of colon cancer, and it's immune cold. And we've really been struggling to make progress in that field. Currently approved treatments include lots of chemotherapy combinations, in combination with monoclonal antibodies that target either the VEGF pathway, which is vascular endothelial growth factor, or the EGFR pathway. But we really haven't made any inroads with immunotherapy, and the reason why should we try immunotherapy in pretty much every cancer is because immunotherapy is what allows you to bring long-lasting benefit. It actually allows us to believe that we can cure cancer. 20 years ago, if we said the word cure in a patient with stage 4 cancer, that would be really unusual. Cancer was seen as a death sentence. My major area of focus in the clinic used to be thoracic oncology. I treated lung cancer patients for 15, 20 years. And so in my early days as a thoracic oncologist, I started in thoracic oncology even before the TKIs came. That's the targeted therapies that target oncogenic driver mutations that I talked about. And so it used to, we could not talk about the word cure, but now I think there is at least 25% of patients on one of the monoclonal antibodies, pembrolizumab, with stage 4 lung cancer are living past the 5-year mark. We generally think of that as being definition for a cure, at least, if of course late recurrences are always possible. And in blood diseases like melanoma, 60 to 70% of patients are being cured with combined immune checkpoint inhibition. This is stage 4 melanoma. And the other cancer where we've really made a huge impact with immunotherapy is kidney cancer, where we've just seen very recently the 4-year survival data from the combination of nivolumab and ipilimumab. So this cure was not possible with all of the wonderful therapies we had before immunotherapy came along. Targeted therapies are wonderful. Chemotherapy is wonderful. We don't want to hate chemotherapy because we are where we are today because of the invention of chemotherapy, right? It's the foundation. It still is the backbone in a lot of cancers. But immunotherapy may, one, provide alternative treatments to chemotherapy in diseases where chemo doesn't work, like kidney cancer or melanoma, or it might provide the opportunity to improve survival in addition to chemotherapy, as in, say, lung cancer. So that's kind of my brief summary on why should we care about developments in immunotherapy in such a wide variety of cancers.
Yeah, I totally agree with you. But at the same time, one should not be not focusing on other TKIs. For example, the perfect example is Gleevec. The first patient who enrolled into the Gleevec trial after 30 years is still now active and alive. And before Gleevec, CML, again, he was given, he was like last stage patient. He was given 2 to 3 months, and he came onto the trial for the first Gleevec trial, phase 1. And now he is in remission for last 30 years. And
absolutely not take it all away from any of the other avenues of research.
Exactly. He has not developed a resistance for Gleevec, which is quite remarkable.
That's unusual, though. That's an anecdotal story. That's
absolutely,
not the common story.
Absolutely true. Absolutely true. Yeah.
I also wanted to make a few comments. It's nice to see that now basic studies are being linked with the clinical trials, and fortunately for biotech companies, development of therapeutic antibodies or now small molecules, what we talk about, I think it has opened up a whole spectrum of opportunity. And unfortunately, I would say our education system has not got into it. We still talk about DNA double helix and Watson and Crick and cloning. I think high time we shift out of it. That's why when I wrote the book for school children, I introduced all that at that time so that they can be taken off at a higher level at much faster, because explosion of information is so much and the approaches are so much that some look very personalized, some looks to a broad spectrum, some looks like a narrow spectrum, just like we had antibiotics. I think it is certainly very, very interesting. And I was very impressed with the ATP story, how people are trying to use small molecules, and the survival rates are happening. So I just want to know, what is the story of P53? Where does it stand? Because it started as a hot stuff those days. Abhay, would you like to make a comment on P53?
Yeah, before making comment on P53, what you said about the school teaching or the college teaching and the, I think, bench to bedside concept or the translational medicine should be given a priority, so that people just don't produce papers and they go into ether and then nobody cares about that paper or publication after 4 more years.
Yes.
Also, I think yesterday we were talking about there are so many papers that are coming out, and then once people start to reproduce that, only 1/3 of the paper data can be reproduced by someone else. So I think academics, fortunately or unfortunately, putting a lot of efforts on the papers and publication, because that's the way to publish or perish. But I think people should start giving a lot of attention to this translational research, translational medicine, so that more avenues open up for students who are graduating from bachelor's, master's, or even PhD level. Rather than focusing on academics only, there should be a lot more collaboration between academics and industry, which will drive the science, which will drive the medicine, which will drive the research on both ends. And patients will ultimately get benefit of that research. And obviously, basic research is really important, but it should not be just done in isolation. Now, coming back to P53, I think initially it was a lot of efforts have been made to make drugs against P53. But what happens is most of the time, almost 50 to 60% tumors are showing P53 mutations. And it's such a common mutation, and the heterogenic pathways are the other pathways don't take into account. It's not considered as a highly targetable or highly effective target for developing the drug. But still, I think the full understanding is not there. A lot of research is still ongoing, and I don't see any specific new hot drugs that are targeted only against P53.
That's true. Another thing I wanted to ask was, what about this cancer stem cells? Everybody is talking. When you do your clinical research with all these markers, are you able to pick up the cancer stem cell from the normal stem cell?
There are two schools of thought. Some people are saying that yes, there are. And one of the Ira Weissman, he believes in cancer stem cell a lot. He founded a company called 47 Inc., which was basically focusing on CD47, where I was working also on that drug. But he believes that, yes, there are cancer stem cells. The others, it's still to be in infancy, and I'm not that much close to the basic research, so I cannot comment more than whatever I've known so far. But maybe some other, or maybe Deepa knows a little bit more from the basic scientific point of view here.
Yeah,
I'm a clinician, so I can't say I can add to the basic science, but what we do know is I can tell you about clinical trials that we've done that have so far been unsuccessful. Whether you believe there is a cancer stem cell or not, I think we can talk about that. That's a whole topic in itself. There are biomarkers that are believed to represent the stemness of cancer stem cells. There are some cancers where stem cells are supposed to explain why they are so difficult to treat, even though a classic example is small cell lung cancer, where you can give good old chemotherapy, cisplatin, etoposide, and you can actually get the tumor to shrink and even disappear in 70 to 80% of patients. However, almost certainly 100% of these patients have the tumor relapse very quickly, within 3 to 6 months of the end of chemotherapy. And the belief there is that, as many of you may know, chemotherapy targets actively dividing cells or DNA replication process. And so cancer stem cells are in a state of dormancy. They are not actively dividing. Therefore, they escape the harmful effects of chemotherapy. And then once you stop the chemotherapy, because patients often, this kind of chemotherapy, patients cannot be on it indefinitely. It's too toxic. And so we usually stop the treatment after 4 or 6 rounds of chemo. Then the stem cells start growing and the cancer relapses. So there have been clinical trials combining chemotherapy with drugs that block the growth of certain proteins in cancer stem cells. One trial I was involved in was with Notch inhibitors. In combination with chemotherapy. I worked on drugs that target HSP90, which targets the chromatin also, but believed to be more so in the cancer stem cells. None of these approaches have been particularly helpful in these cancers. Another cancer where stem cells are believed to explain recurrence and progression is glioblastoma. pancreatic cancer. I think those are some of the more refractory, difficult to treat cancers where stem cells are believed to be responsible for the intractable nature of the disease.
Now, what about this epigenetics business? I mean, will you be able to do some epigenetic modification and suppress that mutation to do a job? Are people looking at cancer approach using epigenetics?
Yes, actually, there is a company called Constellation which just got acquired. They were doing a lot of epigenetic markers, BET inhibitors, for example, that came to my mind right now. But yeah, there is a lot of active research going on in epigenetics there, and people have been trying to exploit that approach also for therapy.
So, if you were to summarize, what are the various approaches broadly? For example, you say surface marker was one. Then you are talking about small molecules which will go inside the cell and block. Uh, uh, you're also talking about epigenetics. And now, what I want to know is that if you were to broadly classify the approaches, so that a youngster wants to get into this area, what are the possible areas where they can go and take a risk?
Yeah, I think the combination approach is ultimately going to win because there are very few drugs where single or diseases where single mutation is responsible for the full cancer.
Okay.
If that's the case, so Gleevec and GIST is a perfect example, or another example is called for systemic mastocytosis, where single mutation D816V in the KIT gene is driving the cancer. If such is the cancer, then obviously single drug will work, but most of the cancers are not falling into this category. Most of the cancers use diverse pathways. So if you block one pathway, the other escape mechanisms start working, and then the uh, cancer escapes. So I think the best approach is going to be the combination approach. And again, one has to see which combination and in what setting. So it's really a complex, and I don't think there is any magic bullets, so to speak, for approaching one way versus the other. But I believe that a combination approach is going to be the ultimate winner. And sometimes, for example, in multiple myeloma, there are 4 drugs are given at the same time, or now people are talking about even 5 drug combinations. And these 5 drugs or 4 drugs will act on
Different targets.
4 different ways. Yeah, exactly. So you're trying to basically block all the pathways in 4 or 5 different directions to control the growth.
Okay. Now, I just want to also ask, methotrexate was the first one which was tried in the, anti-folates. Where is the status of that kind of biochemical approaches? Because everybody is talking about mutations and all that. Are people using basic biochemistry, metabolism? I could see it in ATP, but in general, how many people, are people interested in the classical biochemical approaches?
Yeah, I think actually with the use of artificial intelligence or machine learning, now people are getting a lot of attention to basically put everything into the sauce and see what comes out.
Okay.
And with that approach, I think a lot of new biochemical pathways are being looked at again. So for example, NAMP pathway, adenosine is one of the other example. So understanding of basic biochemistry is really a key. And yes, I think one should not forget to look into those when developing drugs and doing the clinical research or even the basic research. I completely agree.
But what are the model organisms? Nowadays, people don't use model organism to study, create a tumor in an animal and study the progression. They directly
Yeah,
get
I think
on to patient.
right. So what happens is PDX models, patient-derived xenografts, or a lot of times now people are doing organoids. So basically growing cancer in organs in the cell environment, but still those are still in infancy.
Okay.
And one of my colleague basically says all the time, like, I don't care if you cure mice, we have to cure human beings.
Human being, correct.
So and you have seen such data so many times, like, okay, with mouse or rats and guinea pigs, or even in at higher
Monkeys.
species, like when you move on to even dogs and sometimes even in monkeys, macaques, or cynos, you can see the beautiful effect. But as soon as you come to first in human studies, you are not able to reproduce that kind of efficacy. So
Okay.
the system is so complex that ultimately I think the human clinical trials is the key, and then we have to look at the real patient data when they start getting so yes, model organisms or PDX or organoids are useful tools. One can get indication. Obviously it's not going to be the ultimate kind of approach that one can take.
I just have a last question. How popular is bone marrow transplantation for making a disease free? Like thalassemia and all, they do it, or sickle cell anemia. But some of the cancers, blood cancers, is still BMT very popular?
Yes, so actually in AML,
Hmm.
if the patient is still fit, and the fit definition varies, but usually if it is a fit patient 55 years and younger, bone marrow transplantation is tried, and a lot of BMTs are happening. So one of the company is now trying to basically design an agent which will be useful for bone marrow before they do the bone marrow transplantation, to get rid of all the blast. So that they have developed radiotherapy actually.
Okay.
So using radiotherapy with specific targeted antibody. So with the antibody, it's a antibody radiation conjugate. So
Okay.
with the antibody that is specific for CD45, for example, in AML, CD45 is highly expressed. So what they have done is they have created a highly targeted antibody for anti-CD45, and that antibody is tagged with radiation, with alpha radiation, for example, or even with iodine 131. And that basically goes and binds to all the blasts
Okay.
to remove all the blast, and that removal of all the blasts basically conditions patients for bone marrow transplantation. So when the transplant is done, there is 100% grafting, and relapse rates are really, really low. So yes, bone marrow transplantation is still being done, and there are a lot of ways to basically condition the patient before they go on to a bone marrow transplantation.
Okay. Nitin, take over if there are any other questions.
Sure, sir. Other questions, suggestions, comments from the other participants?
I was just wondering, what's the turnaround time? You mentioned some of these. You said Gleevec is good one. You said, right? Gleevec is still a good one. What's the turnaround time for some of these ones that you talked about? What would you say?
Turnaround time for what?
For it to come from, let's say, a molecule to market to be tested.
Oh, it's a really good question. It depends. That traditionally speaking, it used to be anywhere between 6 to 9 years, but nowadays 3 year or sometimes 2.5 year I have seen from phase 1 to approval.
Achha.
Because the depends is the situation changes depending on the length of the study. If it is a small indication or highly focused patient population, based on 52 patient or 100 patient, one can get approval, and those patients can be enrolled in the phase 1 study. So initially what used to happen is phase 1 used to go for 2 years or 3 years itself, because you have to start at the lowest dose and then slightly increase the dose, and the main endpoints of the phase 1s were basically only safety. But
Yes.
now the things are changing. Now in the phase 1 itself, you are doing the patient selection right from the phase 1. You are starting at the efficacious dose, which is going to start giving efficacy right from the beginning. If you know the safety pretty well based on your GLP tox studies in animals, FDA will allow you to start at little bit higher dose. And then again, if you select patients population based on the biomarker, one can start seeing efficacy. So in that case, instead of running phase 1 for 2 years itself, you are running the phase 1 for safety and efficacy, and then doing expansion once you start seeing efficacy based on 20 or 50 patient. If you enroll phase 2 or expand the phase 1 itself into expanded study, add additional 50 or 70 patients, and then go to FDA for approval and see if they agree or not. So again, these are the different strategies that one has to think. We have to consider FDA's input right from the beginning so that they are on board, whether they will accept single arm study, whether they will accept open label study, whether they will accept phase 1 study as a potential approval as an accelerated path. If you have an orphan drug indication, then yes, they are open for looking at approval path in accelerated basis. And then you have to do a confirmatory study once you get a accelerated approval. So now, because of these various technologies and techniques, drug development timelines have shortened, and the fastest approval that I have seen is for serotonin, for example, ALK inhibitor, or systemic mastocytosis. In 2.5 to 3 years, one can get approval.
Okay.
Yeah.
Yeah.
That's quite impressive.
Yeah.
I mean, we used to hear that 12 years, you discover a drug,
12 years, yes.
You file a patent, and by the time it comes on the market, you hardly get 4 or 5 years of the royalty of the patent.
Right.
Yes.
I think it's nice to hear that now 2 to 3 years, if our luck is good, you can enjoy the patent for the next 17 years.
Yeah.
And then
Yes, of course.
Canada, where we have a lot of generic manufacturers, we want to get a scoop of the market. So we want the time phase to be less. So that'll be good,
Right.
So
No, but then generic market won't benefit by this.
Boston area, there must be, no?
No. You can get exclusivity if it is orphan drug indication, or patent expiration is still long. So generics will not be able to enter even if the drug is approved. You will get exclusivity for your patent for number of
20
years.
years.
But how about biosimilars? I think even if you have a patent,
No,
I think
biosimilar is again original patent fellow has the control.
Okay.
So even if you put a sugar or something and claim it,
Yes, of course.
only in India we do this nonsense of process patent. That is why the American companies are refusing to come to India only for that reason, that we created a loophole by creating process patent.
Process patent.
Process patent.
Which is not,
You tweak the process and you get the same product. Yeah.
Exactly. So that is
So
why you call it biosimilar. You are not saying it's the same.
Yeah. And anyway, antibodies cannot be identical. It's very difficult to characterize
I agree.
residue-wise.
But that is the loophole on which you play around.
Yes.
So,
Yeah.
I think. Any other questions?
Does your company have a co-op program with universities, at the undergrad level, like a 1 semester internship?
Yeah, I think a lot of biotech companies take undergrads or even grad programs, and then 6 months internship co-op programs are available. So yeah, a lot of students should explore that in Boston, Cambridge area.
Here in Toronto. So I was wondering whether, because for 1 semester they can go and get some hands-on learning type of thing,
Absolutely, yeah.
Thank you very much. It was a great,
it was a
I
great
didn't
talk.
understand a lot of the things, but I understood the aspects about those small molecule agonists and stuff that you talked about. A few of those. Yeah, 1 second, I have to go.
Yeah, Nitin. Yeah.
Yes, questions from other participants. Okay.
Looks like there are no more questions.
Yeah, if there are no questions, let us thank Dr. Abhay for such a wonderful lecture and giving us insights about how the immuno-oncology works. Thank you, Dr. Abhay, for such a wonderful talk.
Thank you. It's my pleasure.
Thank you. Thanks.
I must thank on behalf of KK Biotech Abhay for taking us through a journey. At least I enjoyed being a student. Listening to Abhay. It was really an honor and a pleasure, because I'm very happy to see how all of you have progressed in life, and gone into bigger challenges. And Nitin for being the moderator today, and doing an excellent job. And we look forward to it. Next one, probably we are planning to have a talk on CAR T cell technology. Okay,
And
because that is another hot
thank you, sir,
area.
for giving us a good platform right from the beginning, like University of Pune, where you were a teacher at that time. We really appreciated that.
Thank you so much. I'm very happy I'm able to see all of you, and listen to you, because it's always a pleasure to see what journey all of you have taken. So thank you so much, both Abhay and Nitin. And please join
Thank
us
you.
for the next session. Every month we have one session on this. Thank you so much.
Sure.
Yeah.
Thank you.
Kanan and all, it was a great evening. It was a great evening, and I'm happy that I have gone back to academics after so many years. Incidentally, for
He is a
other people,
banker.
I and Kannan and Sunil Mangotra, we are all batchmates of 1975. You may not be knowing; it may be a news to you, to some of you.
Thank you,
Great
Vishwesha.
to hear. Yeah, and Abhay, really a lot of information. Now I'm not directly involved with all this, but the background remains same. The base remains same, isn't it? And
Yeah.
Yes.
you all enlightened it, and it is sprouted now again. It started sprouting.
Yeah.
Thank you so much. Yeah?
Thank
Have
you.
a wonderful.
Bye-bye. Yeah. Thank you.
Thank you.
Thank you,
Bye.
everyone.
Bye.
Bye. Bye-bye.